Some of you here may already know Nick. So Nick actually studied the joint degree in psychology and neuroscience here at the University of Manchester. He was actually the year below me, although it'd be best for nobody to try and calculate quite how old we are. And after that, he went on to do his DPhil at Oxford University. And then he is now currently at Nottingham University, having travelled there via France and Israel and Reading. So without further ado, I'll pass over to Nick.
Thanks, Alan. Thanks for the invitation. This is my first Zoom meeting, so forgive me if I push the wrong button. I've put a link to a short video of Seinfeld, which I love. So if you want to watch that quickly first, there's still a few people joining, so I'll let you, if you just watch that, and then I can skip that bit in the talk because that didn't work.
It's very nice meeting you, George.
Likewise. What are you doing? What? Did you just double dip that chip? Excuse me? You double dipped the chip.
Double dipped? What are you talking about?
You dipped the chip, you took a bite, and you dipped again.
So?
That's like putting your whole mouth right in the dip. Look, from now on, when you take a chip, just take one dip and end it.
Well, I'm sorry, Timmy, but I don't dip that way.
Oh, you don't, huh?
No. You dip the way you want to dip. I'll dip the way I want to dip.
Give me the chip!
Hey! Get off!
Double dipping and the problems in analysis and data that I'm going to talk about today. It's a really old problem and it keeps coming up, and I've written papers on it and I've tweeted about it and over the years, and it never goes away. It's always there in the background, and I'm kind of intrigued why that's the case, why we haven't fixed this problem yet after 134 years. So I'm going to explain what I mean by double dipping, and then I'm going to give you some examples of double dipping, and it's pretty relevant these days. And I think the struggle of George and Timmy at the dip counter really highlights the essential tension at the heart of modern psychology and neuroscience. So George is more liberal. He's saying, I'm going to dip the way I want to dip. Whereas Timmy is much more conservative. And he's saying, no, there's a right way to dip. You've got to do it this way. You just take one dip and then you end it. And these two approaches, I'm sort of likening to the way people analyze their data. So George is more of an exploratory, dipping and redipping and having a go at different kinds of analysis, whereas Timmy is much more, no, you do a confirmatory data analysis right from the start. So the dip is the data, and the chips are different kinds of analysis. So the question is, what's the right way to dip? Is George's approach correct? Is George's approach correct, or is it more like Timmy that we should be following? So I'm going to try and illustrate this problem by looking at some old papers, and I've chosen older ones in the 4 case studies that I'm going to talk about to try and avoid embarrassment in case some of the authors are still around and still listening to the talk. The reason I talked about it, the reason I want to talk about this today is because I've done a course this term on the history of psychology, and I added a lecture at the end on the replication crisis and problems of data analysis and double dipping and hacking and harking, all the kind of questionable research practices that have been coming up in the literature, on social media, and even in newspapers about this sort of problem that psychology and neuroscience has in how it analyzes data. So harking is when you sort of change your hypothesis to fit your data post hoc. Double dipping, which is what it's all about today, is doing 2 analyses where the second one is biased by the first one. P-hacking is a whole load of methods to try and get a p-value below 0.05 so that you can publish your work. Questionable research practices. This sort of covers the whole lot of things about things we really shouldn't be doing to our data, but we do them just to get publications and fame. And all of this really contributes to this replication crisis, this failure to replicate modern psychological science. And during this course that I was giving in Nottingham this year, I came across this book by Barber, 1976, and it basically talks about all the same stuff. So all of this replication crisis happened with social psychology in the 1970s, looking back at some studies from the 1960s. So there's nothing new about all of these problems, which I found quite interesting, because we're sort of focusing on this replication crisis, but it's always been there. And double dipping is a fantastic example of a problem that has always been there and that we still haven't solved. And so I'm going to give you a little history of double dipping. So the first double dip, and I'm going to give you 5 varieties of double dip. I'm going to number them from 0 to 4. And I'm giving Galton a free pass on this one, because when he invented this version of the double dip, he also invented regression at the same time. So that's kind of a good excuse to get away with this dip. Now, Galton was studying everything. He wasn't a trained scientist. He wasn't a sort of trained scientist in the normal sense that we might think today, but he was just going around the world measuring everything and writing papers and sending them to Nature. And one of his papers was on the heights of famous people and the heights of great men, typically men. And he noticed that children of giants, so if you take a giant man, he's likely to have shorter children. And likewise, a very short man is likely to have taller children. And at the time in 1877, he was thinking about evolutionary mechanisms of inheritance and genetics. It was, just after 20 years after Darwin, his cousin had published his great book on the origin of species, and the whole Victorian science world was trying to work out how genetics worked and how inheritance worked. Anyway, so Galton proposed that there were these active genetic sexual selection mechanisms to make it so that very tall men had shorter children and very short men had taller children. So he proposed these active functions, active processes to explain this problem of tall men having shorter children. And he came up with this phrase, the progeny of all exceptional individuals tends to revert towards mediocrity. And reversion to mediocrity is what we now call regression to the mean. And in fact, it's what we call regression or correlation in general. So if you look at this picture from Galton at the bottom right of this print that he submitted to Nature, there's the very first correlation coefficient down there. It's not the same one that we use today, but it was the first time correlation was invented. And so Galton was using it as a way to look at what's this active mechanism that means tall men have shorter children. But it was a double dip number 0, if you like. And the reason is because, and he realized this over the next 10 years or so, he had double dipped. And what I mean by double dip, he had first selected some individuals, some data points, and then he'd looked at those data in a second way, in a sort of selective way. So he had selected the tallest individuals and noted that their children were a bit shorter. And he'd selected the very shortest individuals and noticed that their children were a bit taller. And it wasn't an active mechanism that was driving this. It wasn't some genetic or evolutionary thing which was pushing people to be slightly closer to the average in each generation. It was just random variation. So there's this nice quote from 134 years ago. How is it that in each generation there are the same number of men who range between any limits of height we please to specify? Although the tall men are rarely descended from equally tall parents, or the short men from equally short. The answer is that the process comprises 2 opposite sets of actions, one concentrative and the other dispersive, and they neutralize one another, and the generations fall into a stable equilibrium. So I think we would look back and say, he's found signal and noise. So the 2 processes, one is the signal, so it's the genetic transmission of height across the generations, and one is noise, all the other stuff that affects how tall people are, from random variation, nutrition, disease, whatever it is. So in his first paper, he said regression to the mean was an active process, genetic processes, making sure that the breed is fit and so on. In the second paper, he just realized there was signal and there was noise, and every time we select a tall man, we're selecting a tall signal, but also tall noise. And so those two processes always need to be taken into account. So I'm giving him a free pass. I'm not going to label him as a double dipper because he invented regression by doing this, and that's not a bad thing to come out of it. We all like a nice chart showing that things go up. So here's an example of some data from Ngram Viewer Google that shows how many times the term regression to the mean has been used in history. You can see it goes up, as you might expect, with the number of books published. But my point here is that the old term for this was reversion to mediocrity. And that's so Galton and his followers and people commenting were using this term, reversion to mediocrity. And the idea that he was talking about was how things in one generation will revert towards the average in the next generation. And we've just replaced that term with regression to the mean now, but it's the same thing. So all the four examples that I'm going to talk about today are examples of this regression to the mean, of this selective analysis. And it's now recognized as a bad thing. And I'm going to show you some case studies. There's a good paper on this from Kriegas, Korte and colleagues, 2009, and they call it circular analysis and double dipping. But this problem has been around since 1886. So they were just highlighting that this problem in a particular field in fMRI and machine learning. But this problem is everywhere all the time. And when you recognize it, and I'm expecting that quite a lot of you probably will know about this problem, but I'm a bit of an obsessive about this. I've been thinking about this far too much over 20 years or so, and it keeps coming up. And once you recognize it, you see it everywhere, and it's really quite depressing. So today is really a bit of therapy for me to try and get off my chest some things that I've had in my head for a while, and I haven't told anyone about them. So here we are today, a bit of a bit of a confession. So double dipping and this idea of regression to the mean and selective analysis is all about biasing your data by selecting particular things in the data and only reporting those things, or doing other things that might bias the data, and creates these sort of statistical dependencies between your data. And I'm not accusing anyone here of doing this consciously. So I'm not saying this is fraud, and I want to make that very clear because I don't want to go upsetting anyone. What I am saying is that Galton made the mistake and he invented regression. So it's a pretty easy mistake to make. And all of the four cases, they've all been published, and they haven't been, as far as I know, they haven't been criticized for this before. But it's extremely common. And when you see it, when you recognize it, you'll see it everywhere. And that's why I'm giving the talk. So hopefully we can find ways to make this more obvious and identify the kinds of things that people do to their data, which is not good, and hopefully fix it. So there's four examples, and they're all from my research area. So I've just come across them randomly. There's no systematic review being done here. It's just things I've come across over the years, and I want to talk about them. So the first one is about touch perception. Second one about multisensory integration. Third one about body perception, same thing really. And the fourth one about mirror neurons. So the first double dip variety is a really simple one, and this is probably the most common one. And this actually did come from a systematic review. I recently read all of the papers of this research group on a very particular topic. And what they've done, there's an image here of their experimental design. So they've got each of the arrows and each of the little blocks and each of the repeats is the same experiment being done 7 or 8 times. So they establish a baseline, and they do a pre-test and a post-test, and they do that in the first experiment without TMS, without brain stimulation over their somatosensory cortex, and in the second experiment they do it with the brain stimulation. And the point is, it's a pre-post intervention. So they measure touch perception first. They give people this repetitive stimulation, and then they measure touch perception again. And typically they find that people improve in their touch performance after having had this repetitive plasticity-inducing sort of intervention. And in this case, they did the experiment twice, once first, and then 2 weeks later, they did a similar thing. Now, I could do a whole talk on 2-point discrimination, which was one of my options I gave to Ellen, and she went for the statistical one, so that's why I'm doing that. But I could give a whole talk on that, and I will maybe another time. So that's the idea. They have a series of tests, and they have 2 interventions, and they're trying to compare the effectiveness, essentially, of the 2 interventions. And typically, people get better at this task when they have the intervention. It's an old paper, and they didn't put this particular result in a graph, and they didn't even concentrate on this. Is very much a, sort of a post hoc finding, and that's often the case with this variety of double dip, that people just report some sort of post hoc. It's interesting, you know, they're exploring their data. So the expected result was that they found that thresholds improved after the intervention, and it looks like a small number, 1/4 of a millimeter, but in this situation, this is quite a big improvement in threshold. But the important thing, and the thing which raised my hackles and made me aware of the problem, they said this at the end, in the end of the results. We correlated the individual gain after co-activation and TMS, normalized to the gain found after co-activation alone, with the gain after co-activation alone. And this revealed a strong negative relationship, R equals -0.7 approximately, which is significant. So subjects that benefited greatly from co-activation, in one case, didn't show much improvement, and subjects that showed little improvement in the first one showed a large improvement in the second one. And whenever I see a statement like this, my warning signs go off in my head, and I'm thinking about Galton's tall men and short men. So put in Galton's words, this would be just another example of tall men having slightly shorter children and short men having slightly taller children. So subjects that benefit a little bit in the first experiment benefited a lot in the second. Subjects that benefited a lot in the first benefit only a little in the second. So it's the same thing. They're selecting the low improvers in the first experiment and finding that they improve more. So it's regression to the mean. And another thing about this number is 0.7, is that this is the number you get when you when you put random numbers into the equation. So I'm going to spend a bit more time on this particular example, just so we all get the idea of what it is I'm talking about in general, and then I'll skip through the next 3 case studies. So this variety of double dip, the improvements from baseline or the regression to the mean, essentially, it often occurs when scientists report 2 quite complex things. So there's always different scores or sort of quite involved processing of the data. And if you correlate 2 quite complex things together, it's quite hard to remember where the original data came from. And in this case, Raggett and colleagues, that was true. So they had various gains. They had 2 different gains. They had gains relative to each other. It becomes sort of differences of differences, and it becomes quite difficult to remember where the original data come from. And the problem in this specific case is that each of those 2 things that they're correlating actually includes the same baseline data. So in one experiment, it was one condition versus the previous condition, the previous experiment, and in the other variable on this correlation, it was the original one alone. So it was a difference from baseline correlated with the baseline. And when you do that, when you do this sort of analysis, very common, how did my baseline scores correlate with the changes from baseline? You often get, if you put random numbers into this equation, you get a significant negative correlation, which happens to be the same as 1/√2. And there's probably a mathematical reason for that. I haven't looked into the maths, but it always comes out around -0.7, which you'll see from the previous slide is exactly what they found. So -0.7. So when you see this kind of R value, and you see that 2 complex things are being correlated with each other, and that the same data sort of appear in different variables in different ways, you should start being suspicious of this double dip variety. And authors will often write things like, the poor performers got better and the good performers got worse. So it's regression to the mean. And in case you don't believe believe my interpretations. I've got some random number examples to show you and some code that you can use. But I found, and I've always wondered how common this is, and I've never done a systematic review apart from this one. So I looked at all 41 papers from this lab over nearly 20 years, and 8 of those 41 papers contained this variety of double dip. And none of them were really critical to any of the conclusions of the paper. They were just an additional finding that they sort of explored post hoc. But 20% of the papers contained this problem. The fact that they are correlating two things which are actually statistically dependent, and therefore, violating the assumptions of the statistical tests. And I'm going to show you, if I can, with. Can you see this spreadsheet now? Someone?
Yes.
Cool. Good. So it's really easy to find these sort of double dip analyses, and you can just do it in Excel or Calc in this case. So I've replicated their experiment with just two conditions. So there's a baseline score, and then there's an intervention score. Can you see the cursor that I'm highlighting sections? Can you see that, or is that?
Yeah, we can see that.
Excellent. That makes it easier. So you've got a baseline score, and then you've got an intervention score, and then you've got a change score, which is just the difference between the two. How much did they improve in the second one compared to the first one? And you can take means and standard deviations and all that stuff, and you can correlate the two things. So I've got a correlation there between the two things. But when you correlate the baseline with the change score, you get a very different correlation. You get this very strongly negative one. So the original correlation between the two data sets was minus 0.17, and the double dipped correlation is minus 0.8, which is significant. So I've done a t-test there on those data. And if you compare the top and the bottom graphs, the top graph is the correlation between the raw data, the actual original data they tested, and the bottom one is the double dip, the comparing the change from baseline with the baseline. And as I just refresh these data, you should be able to see that the p-values are almost always significant, and you almost always get a strong negative correlation, which looks like a real thing in the data. So if you start doing this kind of double dip, you'll see this plot and you think, oh my God, I found something. It's really interesting. It's a strong negative correlation. That's very interesting. But of course, all of these things are just random numbers. So there's no effect being simulated here at all. It's just sticking random numbers into the analysis pipeline that the authors did. And that's a problem. So it may be the case that there really is a true effect in their data, but because they didn't check to see whether their analysis was not biased, you can't really tell what they reported was actually real or not real. Is it significantly more or less negative or positive than what you would get from random numbers? And so it's very easy when you see this sort of description just to create a load of random numbers, simulate 12 subjects, simulate 2 conditions, and just repeat the data analysis and see what happens. You can do it in a spreadsheet there, or you can do it with code, of course. It's really easy. There's only a very few steps. And I've put my code here on my web page. And what I've done in this code is really simple. So I've just repeated the analysis 10,000 times, and 10,000 is a good number for these sorts of simulations. So here's the pseudocode. For each of 10,000 simulations, you just generate random numbers for each trial, condition, and subject. And then you just do exactly what the authors did, do the same steps that they reported, and calculate the same statistics that they reported. You do that 10,000 times, you can get a histogram then of the statistics you get from just repeating this analysis. And if the histograms are biased away from 0 or whatever the expected value is, then you've got a problem. And this is the kind of thing you get. So I did 10,000 times of this, just random numbers, looking at how the baseline correlates with the change from baseline. And you can see that all of these distributions are heavily skewed off of 0. So in these graphs, I'm going to show you more of these graphs later. And in these graphs, the black line in the middle is the expected value. So it's usually 0 in this case. The red line is for the critical value, what would be significant, statistically significant at 5%. And then the black data are what you actually get from this kind of analysis. And so you can see on the left graph that the R values are heavily skewed towards negative numbers, and they're around -0.7. And so most of those R values will be significant. If you convert the R into a t-test, you get slightly more symmetrical distribution, and the t-test slightly more easy to do stats on. And we're getting 82% false positives. So with this kind of analysis, rather than 5% false positives, you're getting 82% false positives, which is pretty bad. And then on the right graph, you've got the distribution of p-values, and of course, heavily skewed. So most of the p-values are significant. And I've cut off the graph there. It's actually very tall. So it goes almost, 82% of the data falls below the significant level. So it's a very simple thing just to correlate the change in a score with the original score. But you're going to have, depending on the sample size, very large numbers of significant, apparently significant false positives data just from doing this sort of data manipulation. This is at the end of the first example, which I took in detail. Are there any questions about this now? It might be a good time to take any quick questions about anything that wasn't clear before I push on. If you want the code for free, you can go to my webpage, and then this is the entirety of the code. If you understand MATLAB or Octave, then that might make sense to you. If there are no questions, which I don't think there are, that's good. I'll just carry on. So the code is very simple. It's very simple just to simulate these data sets and just show that people are creating significant effects where none really exist. And without doing that on the original data set, you can't really say whether the original data is really significant or different from what expected, or if it's just the same as random numbers, essentially noise. Is there a signal or is it just noise? So look at the noise. Look what happens when you put noise into your analysis, and then you'll find out. Right, so that was the first example. I'm going to go through three more, and they're all very simple. They're all basically variations, slightly more complex each time on the basic theme. So this field is what actually got me started into double dipology, and this is number 2. This is something called inverse effectiveness. That's why I've called this variety the inverter. But essentially, it's the same problem. It's just slightly more complex. So these now are, it's a paper in Science in 1983. They studied 108 neurons in the superior colliculus of a cat, I think. And the colliculus analyzes and integrates multiple sensory and motor signals. So auditory, visual, tactile. If you're a snake, your infrared pits from your face projects to the superior colliculus. And then all colliculi then integrate these sensory signals and produce eye, head, and limb movements. So it's a really beautiful model for understanding sensory motor integration, and it has been a model for probably 50 years or so. The particular finding that I'm focusing on is this idea of multisensory integration. So what happens when a cell in the colliculus receives both auditory and visual stimuli? How does it integrate those two stimuli? And the classic story is that you may have a very weak response. So in the diagram on the left, you've got A, B, C, and D panels. In panel A, you present an auditory stimulus, and the cell responds a little bit, a couple of spikes per trial maybe. You present a visual stimulus, and this is panel B, and the cell responds a bit more, 2 or 3 spikes per second or per trial. But if you present them both together, auditory and visual, at the same time in the same location, you then get a very vigorous response, so 10 or 11 spikes per trial. And this is the classic finding that unisensory stimulations can each have their own small response in this cell, but you combine them and you get a massive enhancement of the effect. So they studied 108 neurons like this. 26 showed enhancement, 21 showed a significant suppression, so the opposite sort of effect, that when you presented auditory and visual together, the overall response would decrease relative to the best unisensory response. And 42 cells showed nothing. And this paper was really the start of a massive field of multisensory integration research. And it's sort of the paradigm of research. And from the original paper, Meredith and Stein talked about when individual stimuli were just above threshold for eliciting discharges, the enhancement was greater than when the individual stimuli were highly effective. As the effectiveness of the individual stimuli increased, the percentage of enhancement decreased. And this has been called the principle or the law of inverse effectiveness. And of course, you can think that this is just like Galton's tall and short people again. So very effective stimuli in one condition show less effectiveness in another one. So very, very weakly effective show a strong enhancement, and very strongly effective show a very weak enhancement. And I've written various papers on this over the years. And it's important to note that it's just, so if you just put random numbers into these equations, you'll get the same thing. You'll get that things with very low responses in one condition will tend to have a higher response in another. But also there's floor and ceiling effects. So in the case of neurons, you can't have less than 0 spikes per trial. So it'll just be 0. It'll be a non-responsive neuron. You won't even study the neuron. If you don't get any responses in a particular condition, you won't study that neuron further. You'll just remove it from your data set. So in fact, there's these floor and ceiling effects and selection effects operating that in order to be defined as a neuron that's worthy of study, you have to have a particular pattern of responses. And those patterns in this case are, in the case of a neuron with a very low response in the unisensory conditions, they have to have a much higher response in order to be called a multisensory integrator or a multisensory enhancer neuron. So there's a bit of selection going on. This is why it's slightly more complex than the first example. There's a bit of selection and a definition and a classification of responses in one way or another. And of course, you can stick this into your double dip calculator. So I've just done what they've done. I've got 108 neurons. I've got a visual response, an auditory response, and an audio-visual response. So now there are 3 conditions rather than the original 2 that we saw in the first dip. And the way they calculate the responses is by looking for the maximum unisensory response, so the maximum of the auditory or the visual. And then they calculate a measurement of multisensory integration based on how the audio-visual condition differs from the maximum of the auditory or visual. And I could talk about different equations for doing this, but most of the equations come up the same sort of problem. You can calculate a value of multisensory enhancement for each of these neurons, and you can take the logarithm because it's a sort of a power law because there's a division going on. So you basically do the same kind of analysis that they did. They've calculated multisensory integration. They've selected the neurons which are showing an enhancement, and then they're looking for relationships among those neurons. And you can probably guess that these are all just random numbers. And if you put random numbers into this equation and do the selection that they did, you always get a very strong, highly significant power law. And this looks really convincing. And they've plotted graphs exactly like this in their papers over the years. That there's a power law that relates the amount of multisensory integration with the strength of the best unisensory stimulus. And there's a similar law if you look for multisensory suppression rather than enhancement. But this is just random numbers, and it's almost always significant in a t-test. And you can just play with the random numbers all you like. As long as your sample size is big enough, you'll always get a really nice relationship. So my conclusion when I gave a talk about this in 2008 was that this isn't a property of multisensory integration, it's a property of multisensory equations. They didn't like that when they gave that talk. So you can do it with just random numbers in an Excel spreadsheet, or you can do it with the code. It's just a slightly more developed code than the previous one. And this analysis, because they have 108 neurons, just with random numbers, you always get a very highly significant relationship, negative correlation coming out. So the R values are highly biased to negative numbers. We've got nearly 100% false positives in this case. An extremely high proportion of analyses will give you a significant relationship where none really exists. So it's a property of the multisensory equations and the processing. It's not a property of the original data. And again, I'm not saying that the superior click list does not integrate multisensory stimuli. It does. I've done studies like that myself. It definitely does integrate them. What I'm saying is this law of inverse effectiveness has not been shown to be any different from what you'd expect from quite random variation among response levels. And this is not just one example from 1983. There's a whole literature on this, probably dozens of examples. And there was one, it's the bonus track in this talk. There's one in 2010 that I saw was a problem, and I wrote a commentary on it, and I simulated the data and I sent it to Current Biology, and of course they rejected my commentary, even though everyone agreed that this, even though everyone agreed that there was a statistical problem. They didn't allow me to even publish it as a commentary online, not even as a letter. Still bitter. In this graph, if you look at the middle panel B, you've got the real data from monkey local field potentials in auditory cortex. That's the real signal, the underlying signal. And then you've got some simulated data that I made on the right side of the figure. That's just by adding together some random sinusoidal components, just to approximate what the signals looks like. Figure 1 of this Current Biology paper contains the bottom left graph. That's taken straight from the paper. And so they were saying that these 3 classes of neurons were fundamentally different from each other. And on the bottom right of the paper was the data I recreated just by putting random sinusoids into the same analysis pipeline that they reported. So it's not just individual neurons. You can do this with local field potentials. You can do this with any kind of data at all. So don't, yeah. Just because something's complicated doesn't mean you can't sort of recreate it with random numbers, and which is what I did. And unfortunately, they said they were aware of these problems from my previous work, but they still refused to correct them, which is interesting. Let's move on. The third example is quite a common one as well, slightly more complex than before, but not much. This is a paper on, again, using TMS to look at touch and processing of the body. So in this experiment, participants look at an object, it's either the hand or a piece of wood, sort of an abstract shape. They then get a TMS pulse over their brain, somewhere up here, and then they get a stimulus, a target stimulus, and the stimulus is either oriented across or along their fingertip, and they just have to say what's the orientation of the tactile stimulus. And the typical finding in this field is that when you look at your hand, your tactile stimulus, your tactile perception gets better. And what they showed, if you look at the top graph here, they showed that when you compare the effect of viewing a hand stimulus, either with or without TMS, so that's the 2 bars on the top left of this graph. The white bar is bigger than the black bar, and that shows that TMS in the black bar has removed this visual enhancement of touch, what they call it. So in the typical case, you get a better performance for viewing the hand than viewing a neutral object, and that's the 2 white bars compared to each other. But when you apply TMS, there's no longer any difference between these 2 bars. And that's what the whole paper depended on, this difference being significant. They repeated it in a second experiment. So the second experiment at the bottom of this graph, it's the same thing. They just did TMS over 2 different areas. And they found that TMS over S1, primary somatosensory cortex, got rid of the visual enhancement of touch effect. But TMS over S2 didn't. And they made conclusions about S1 being more of, S1 was the source of this enhancement effect. And that seems fine. It sort of fits with the literature, and it fits what you might expect about S1 and S2. And there's a but. The problem in this paper, which jumped off the page when I read it, was that participants were selected for showing the visual enhancement of touch effect. And that's this contrast at the top, the two yellow contrasts there at the top. So in order to be included in the analysis, you had to show the visual enhancement of touch effect. And this is like the responder or non-responder analysis that you'll see in various data sets. And in the first experiment, it was only 3 out of 11 who didn't show this effect. But in the second experiment, it was half didn't show this effect. And these numbers are pretty stark. So in the second experiment, they removed half of the subjects who didn't show the effect. And to me, that sounds actually like there is no effect at all in the population. And in reality, so if you just remove the subjects who don't show the effect, you're going to end up with a biased data set. That could be okay. It might be fine to just study responders. The problem is they left these data in the analysis, and their conclusions were based on comparing, for example, the hand condition without TMS to the hand condition with TMS. And so by selecting participants in a first stage, they're then biasing the second stage if they leave those same data points in the analysis. So the selected data showed significant effects, and they drew conclusions about that. And the unselected comparisons showed no significant effects, and they drew conclusions about the lack of significant effects. So same deal with the spreadsheet. You can get now there's 4 effects rather than 3. There's 4 conditions, A, B, C, and D, and they're all random numbers. And you can select people according to whether they show a particular difference between columns A and C. You then do a selective analysis just on those data points that you've already selected. And then you can just repeat the analysis as much as you like. And the key thing for their paper was that the difference between A and B was significant, whereas the difference between C and D was not significant. And it's because the sample sizes are now very low. It looks these data look quite noisy, but you still get the same basic effect that each time you run the data with random numbers, you get the reported significant effects. So if we do the proper thing and look at a simulation with 10,000 data sets, so because the original sample was quite small, and they selected sort of half of those subjects to continue in the analysis, the distribution of these effects is much wider, and you get fewer false positives overall. But that's just because it's a small number of subjects. All the biases are all there. So in the top 2 graphs of this figure, they show an effect which has been biased by the pre-selection, and they've got 25% false positives. And in the bottom 2 graphs, that's the comparison of an effect that wasn't selected for by removing some participants. And that you can see is symmetrical, and you get the expected number of false positives, 2.5% in each tail. So in this paper, they selected subjects based on a comparison between 2 conditions, but they left those 2 conditions in the analysis. So any comparison which included either of those conditions was biased, and the one comparison which didn't include either of them was not biased. And that was the only fair comparison in the paper, and it's the only one which wasn't significant. So there's that. In the final 5 minutes or so, I'm going to give you a very high-profile example from the mirror neuron world, and this is even more complex than the previous one. So when you read this one, it's quite hard to pick out that there's a problem. So mirror neurons are neurons in the premotor cortex which respond to observed actions as well as executed actions of a monkey. And in this paper, they selected mirror neurons from the ventral premotor cortex, but they also tested the distance sensitivity. So they moved the target stimulus at different distances from the monkey. And so they found cells in their 105 cells, they found 27 which preferred a far stimulus and 28 which preferred a near stimulus. And before I carry on, I think you've all been trained now to be expert double dipologists. So can anyone see a problem with this figure? Either of the graphs, so either of the red or blue graphs in the bottom of this. Does anyone want to volunteer what might be wrong with this data set? Anyone want to be brave? Get a mince pie if you're right. Okay, maybe I'll just tell you. So when I saw this graph, I saw immediately that there was a problem. And it was the size of the error bars. So if you look at these data, the error bars, there's 5 distances being tested on the x-axis, and there's the normalized activity of these mirror neurons on the y-axis. And for 2 of the 5 positions, the error bars are tiny, really very small. And the other 3 positions, the error bars are quite big, much bigger than the others, right? And that's the same for the extrapersonal neurons, the neurons that preferred stimuli in far space, as well as the peripersonal neurons, the neurons that preferred stimuli in near space. And so I saw this and I thought, oh, error bars problem. Let me have a look at the data. And the data is quite complicated. The data analysis is quite complicated. There's various stages of selection and processing and normalization. But essentially, you've got 5 distances. So it's a slightly more complex spreadsheet here. I don't expect you to read all these numbers. What they've done, they've taken data from 5 different distances. They've classified the neurons according to preferring far or preferring near. They've then checked that there's a significant difference between those 2 conditions. They've then selected those cells. They've then normalized the data, to fit into the same range, and then they've summarized the data. So you can see there's a lot more processing going on here. So when you read it in the paper, there's like 4 or 5 steps. But essentially, if you just repeat the data analysis steps that they reported with random numbers, you get graphs that look like this. And they're not perfect, but if you just try a few iterations, occasionally you'll see a graph that looks pretty similar to what they reported in their paper. So if you saw these 2, for example, I just press refresh a few times, you saw these 2, you'd think, oh, there seems to be a relationship there. But these are all coming from random numbers that have been selected for showing what the authors were trying to show. And we can, of course, look at the expected distributions of statistics under random variation. And in these analyses, you get about 50% false positives for the relationships that they were talking about in the paper. So in this last case, I think this paper falls on this claim. Yes, you can find cells that have a distance sensitivity for near versus far, and you can classify them, but there's no evidence in this paper that they show a sort of a tuning curve of distance. Because of the way they selected and processed their data. And this is my cute comparison of, at the top of this figure is the data, the charts you get from just generating random numbers and doing this 10,000 times. And at the bottom is the data they showed in the paper. Now, again, I'm not saying that there isn't a distance selectivity in the real mirror neuron data in this science paper. What I am saying is that the true baseline, the true null hypothesis is illustrated in the top panel. That's what you expect by chance. And the bottom panel is what they found in the monkey. So the true statistical test is whether the top panel is different from the bottom panel. It's not whether there's a slope or any sort of apparent function in these data. That was a pretty high profile example. So for all of these examples then, and Krieger-Skorter points out all these sorts of things as well from another field of analysis. And I think the best way to summarize it is in Krieger-Skorter's words, that double dipping is selecting data and then selectively analyzing the same data. And it occurs in many, many different ways. I've showed you 5 ways, but Krieger-Skorter and colleagues talk about maybe 5 more. It happens in every field. It happens from 1886 all the way to 2019. I keep seeing it all the time, and it sort of niggles away at my brain until I write a talk about it. So it's selecting data, and that could just be by your eye. You could just look at the data and think, well, that's an extreme example. Let's see what happens with that extreme example. So you're selecting with your eye, and then you're doing a selective analysis. You're looking at what changes. You've got to take into account the fact that you selected in the first place. And if you don't take that into account, for example, by putting random numbers in, then you're going to get biased results. I don't know how common this is. My guess is probably in 10% of all papers, I would guess. And there's absolutely no basis for that guess apart from sort of a gut feeling. So what can you do about all this? First is finding the problems. So learn about what it is to have regression to the mean, really understand it, and then I think you will find it everywhere. You'll find it in politics. I've seen it there. That there was a conservative policy on free schools about 5 years ago, and they basically did a double dip, and there was a regression to the mean, which made its way into national policy, which is interesting. So it's everywhere. It's in politics. It's in science. It's in anywhere you've got numbers. And some of the things that stick out to me when I'm reading is if the error bars are unexpectedly small, maybe for some conditions, or just they don't look right. Always check how big the error bars should be. or maybe if the error bars are too big. So if you're used to a particular sort of variation in your kinds of data, and you see these massive error bars on comparisons, which you expect to be much smaller than the difference between conditions or the difference between subjects, then again, that's a possibility. It raises the possibility that there's some sort of selective processing going on. Authors who double dip will say things like, we classified responses like this, we selected, we normalized, we removed non-responders, and then looked at what happened, looked at what was left over. That's the kind of thing they tend to say. And if things look too good to be true, then it probably is, and you should be a bit suspicious. And I should say these are only heuristics, of course. I'm not saying that every data set with small error bars is bad. It should be just, you need to calibrate your expectations about what the real variability in data is like. So once you've found the problem, you can try and fix it by just putting random numbers into your data set, just to see if your analysis pipeline ends up with a biased distribution of statistics or not. You can simulate the analysis 10,000 times and plot the distributions. That's probably the most useful thing to do. Another approach is to divide your raw data into 2 sets or more, and you do all your fishing and exploration on one data set, and then you do all your confirmation and hypothesis testing on the second independent data set. That approach is almost universal now in machine learning sort of fields. Or you could get some sort of other independent data. Could be another experiment, another researcher using a meta-analysis, whatever it is, you need some independent way of setting your analysis parameters. And this problem is real. Only last week on Twitter, a very famous scientist, Sam Schwarzkopf, retracted a paper from Current Biology because he found in his population receptive field paper in Current Biology that they'd basically done a double dip over multiple experiments and multiple time points, and they did the right thing and they've retracted it, and I thought it was a real brave move. We should really celebrate. But it's extremely common. It happens all the time, and it is definitely more common than you would hope. Right, to finish, what would Francis, Timmy, and myself say about this problem? Well, when Francis Galton realized that he had double dipped and he invented regression, he felt a glow of loyalty and respect towards the magnificent sway and sovereignty of his mathematical analysis. And he had to smooth his data with tender caution. So I think we should all be smoothing our data, but do it very tenderly and very cautiously. Timmy would take an extreme view and just say you take one dip and end it. So he's saying just do confirmatory hypothesis testing only, don't do any fishing or dipping at all. And I would say, actually, you can dip as much as you like. You can take 2 or 3 or 4 dips, but it's your responsibility to make sure that what you're doing is not biasing your statistics. So you must model, or at least simulate, or just check with random numbers, whatever you're doing. I think I'm done.